Hi, my name is Lo Clark. I will be demonstrating the gram stain and heat treatment in this video. Unfortunately, I've done this video a couple times and I've realized that my little guys are going to be backwards on here. I thought that it would show forward if I wrote it backwards. Technology is a little more ahead of me than I thought. Oops. Anyways, procedure-wise, materials we're going to need: protective. We're going to want gloves for our hands, goggles, and a lab apron or coat of sorts. Materials we are using for this procedure is going to be our Bunsen burner. We've got distilled water, our inoculate loop tool. The media I've chosen is an agar slant media. We've got our bibless paper, a clean and sterilized slide, and then we've got 4 stains here: our primary stain, crystal violet, our mortar stain, grams iodine, our decolorizing stain, acetone alcohol, and our counter stain, safranin. Now, to start the procedure, we're going to do the heat treatment. So we're going to take our clean slide, I'm going to take my inoculate loop, I'm going to run this through my Bunsen burner flame. This is going to sterilize it. Now I'm going to take my water, I'm going to put like a drop on it just to catch it in the loop. Then I'm going to take my clean sterilized slide, I'm going to smear the water right in the center of my slide. Some might have like a wax circle to show where they're going to spread it. It might also be what they use to label the slide for this demonstration. Okay, I'm going to go ahead and re-sterilize my inoculate loop back through the flame. And now I'm going to take my agar slant. I'm going to remove the cap, but without placing it down. I'm going to take the edge of the tube, run it through the flame. This is going to sterilize the edges and remove any air. I'm going to take my inoculate loop and very gently grab a colony out, run that tube back through the flame, put the cap right back on there, and we go. Okay, got my slide again. I'm going to take my loop with the colony on it, and I'm going to smear it right in that little spot where the water is at, nice and good. I'm going to do it until you start to see a film. That's how you kind of know that it's good and mixed. Once you're done with that, go ahead and re-sterilize your inoculate loop. You are done with that guy. We're going to go ahead and let this air dry for about 10 minutes until that little spot is completely dry. Once you've waited, you're going to take that same slide and you're going to run it through the flame 3 times. 1, 2, 3. That's going to heat treat it. That way any cells don't wash off or you lose any just from the steaming and the rinsing off with the water. Okay, moving on to the staining portion. Got my catcher. I'm going to start with the primary stain. That is my crystal violet. Now this is going to color all the cells initially. I'm going to do a couple of drops on there, and that's going to sit for about a minute before you take your water. Turn it around. At an angle, you're going to rinse that off into your little cup or sink. Excuse me. Oh, so sorry. Next, you're taking your more gram iodine. This is going to bind to the primary stain and lock the peptidoglycan wall. I'm going to take a couple of drops on there, let it sit for another minute. Take your water, same thing at an angle. I'm going to go and rinse that off real quick, couple drops. Now we're going to take our decolorizing acetone alcohol. This we're going to do a little bit differently than the other stains. We're going to do quite a few drops until you start to see the runoff just barely start to get clear. And then don't let this one sit. Take your water immediately, rinse that off. Now the decolorizing acetone alcohol, that's going to remove the primary stain from the gram-negative cells. And it's doing that by disrupting the lipopolysaccharide cell membrane. Finally, we're taking our safranin. This is our counter stain. This is what's actually going to recolor those gram-negative cells. A couple drops like the other two. Sit for about a minute. Take our water at an angle. Rinse it off again. Now we're going to take our bibless paper, give her a little dabarooski, and this guy is ready to view under the microscope. Now our microscope has a couple of settings. Our ocular lens is going to be times 10, and then you're going to have 4 objective lenses that are going to multiply that. You got to times 4, 10, 40, and 100. This is going to give us a total multiplication lens view of 40, 100, 400, and 1,000. Now we're going to look at this guy under 1,000, but in order to do that, you need to oil immerse your lens. That by doing that is going to allow you to see enneagram reaction, arrangement of the cells, and morphology. It's also going to indicate a cell wall thickness as well as the presence of a polyliposaccharide membrane. And that is the gram stain and heat treatment demonstration. Thank you for joining me.